So, in the first part of the lectures this morning, I talked about what a cancer is, how we classify those cancers, and the normal cell growth, how we control that, mammalian cell cycle. And again, I would like to re-emphasize that understanding cell cycle and the control of cell cycle is central to understanding how cancers initially form. And I talked about the 6 hallmarks of cancer, and again, just to re-emphasize that, you should be familiar with those hallmarks and be able to explain each one of them. What I'll talk about for the remainder of this session is how DNA mutations impact on cancer development and the stages of cancer and how that links in to survival and prognosis. In terms of learning objectives, understand and explain how cancer leads to increased mortality, so how having an uncontrollably dividing ball of cells in your body increases the risk of you dying. Describe how mutations in oncogenes are a key factor in the initiation of cancer. And this kind of flows out of much of what I was talking about in terms of the early hallmarks of cancer. What you will have noticed is that mutation of key genes, either oncogenes or tumor suppressor genes, are one of the key initiating factors in the formation of cancers. And discuss the different clinical stages of cancer, understanding the link with survival rates. And I'll talk a little bit about our understanding of the underlying causes linked to cancer. So, how does cancer kill? Having an uncontrollably dividing mass of cells within tissues in your body can have serious consequences for the function of that tissue. So if you have an increasingly large tumor growing within a particular organ, it might be your liver, might be your lungs, might be your kidney, might be your brain, this has an impact on normal organ function. So for example, it can block or obstruct the function of that tissue. It can deprive that tissue of nutrients. If you have a constantly dividing ball of cells, that is sucking up nutrients that should be taken up by the normally functioning non-cancer cells. And it can cause pressure. So going back to that image that I showed you in the first lecture of a brain with multiple metastases sitting within the brain tissue, the presence and growth of those tumors within the brain actually can have severe consequences in terms of brain function, leading, for example, to changes in personality as the normal function of the frontal lobes is disrupted by the presence of a tumor in that region. And so there are very serious consequences of having an uncontrollably dividing mass of cells within an organ. It can interfere with metabolic processes, so going back to the deprivation of nutrients, this can result in malnutrition, changes in calcium regulation, alteration in liver enzyme function if it's a liver tumor, production of blood cells. This is particularly true of bone marrow cancers, so for example in leukemia, and in the dysregulation of hormone production, either overregulation or underproduction. And this, in turn, can lead to muscle wasting. I think this is quite well illustrated by this diagram here. So what you have here is a life history of a tumor. You have a normal cell sitting on the outside of a tissue. These cells undergo oncogenesis and become cancer cells, and eventually develop into a tumor. They abnormally multiply, deriving from those 6 hallmarks, and dysregulation of those 6 hallmarks that we talked about in the first lecture. And eventually, that tumor will become malignant, or some tumors will become malignant or invasive. And instead of just sitting on the outside of the tissue, it will actually burrow into the tissue, disrupting and damaging the normal function of that tissue. And this is shown more clearly here. So here we have the primary tumor. There's a local invasion where the cells are moving and burrowing down into the tissue, impacting on lymph vessels, promoting angiogenesis, so increasing the blood supply to the tumor at the expense of the normal tissue, and eventually leading to metastasis, where the cells break off and go into the bloodstream and move away to other sites within the body. In terms of what causes cancer, And again, this really goes back to those initial events in terms of reduction in reliance on external growth factors and an ability to evade suppression of cell division. Much of this is driven by mutations. So the initiating event in oncogenesis is a mutation in one or other of the oncogenes or tumor suppressor genes. So this, a single mutation, a single change, leading to the acquisition of a property matching up with one of those early hallmarks of cancer, and leading to increased proliferation, that in itself is not enough to lead to cancer. And this is why, going back almost 20 years now, those 6 hallmarks were developed. So just one change is very unlikely to lead to the development of cancer. What happens is that a single cell, as it undergoes a transition from a normal cell to a cancer cell, undergoes multiple mutations, affecting a number of different genes, both oncogenes and tumor suppressor genes, and also some of the other genes that I talked about in the first part of this lecture. Developing or matching up with most or all of those 6 hallmarks in order to progress to cancer. And this really is why understanding those 6 hallmarks is so important. Altering one of those is not enough to develop cancer. In order for a cell to form a tumor and to spread, actually it needs to undergo changes relating to almost all of those hallmarks. And this takes a long period of time. And one of the key thing to note is that the incidence of cancer increases the older you are. This is partly because it takes a long period of time for those mutations to accrue and for a cell to undergo the change from a normal dividing cell to a cancer cell. Of course, it should be noted that there are also childhood onset cancers, quite often driven by underlying mutations in the genome that dysregulate a number of processes from birth. But in terms of incidence of cancer, and I'm talking about cancer globally now, across all of the different types of cancer, this increases as time goes by. This is also linked to an increase in mutation rates and a lack of control of the DNA replicative process as you age. So what causes all of this? So DNA in a typical cell is damaged many, many times a day. So in each individual cell, there will be multiple incidences of DNA damage. This is part of a cell functioning. The cell is a very dynamic environment and there are a lot of chemicals present in any given cell which can cause DNA to be damaged. So in a typical cell, around 10,000 times per day, an incident of DNA damage will occur. Now, much of this DNA damage is repaired, and actually our cells have evolved quite sophisticated mechanisms to detect that DNA damage and to repair it. And actually P53 is a detector of DNA damage, is an example of this. The BRCA1 gene, which is linked to risk for breast cancer, is also involved in that process. Unfortunately, these mechanisms are not perfect, and occasionally some damage is missed, leading to a mutation that is passed on from cell division to cell division and enters into the genome. These mutations occur randomly throughout the genome, so actually it's very, very difficult to predict where exactly in the genome these will occur. And in many cases, these DNA changes are in parts of the genome that aren't actually part of a gene. They're parts of the genome that don't code for individual proteins, some of which may cause damage. It may alter regulation of DNA, but do not directly affect a particular gene or a particular protein. So these may have no effect. However, when these mutations occur, so what I'm talking about in terms of mutation is a change from one nucleotide to another, or a more serious structural change in the DNA, which I'll talk about in a few slides. When this affects a proto-oncogene or a tumor suppressor gene, this can lead to cancer. And at the heart of this is the structure of DNA. So this is just a zoomed in cartoon of a chromosome showing the DNA sequence that underlies all of the DNA in your chromosomes. And it's a change in that nucleotide sequence coding for a particular gene that results in mutation and is one of the key events driving the formation of tumors and cancer. And these changes can be of different types. So in terms of a gene coding region, a point mutation is an individual change in one particular nucleotide, changing, for example, from an adenine to a thymine. And that change in the DNA sequence can change the protein that is produced. And again, this harks back to a lot of what you covered in part 1 in terms of DNA replication, the central dogma of molecular biology. And if you're not comfortable with the fundamentals of DNA replication, DNA being converted to RNA and then being converted to protein and controlling cellular function through that process, that is a very important concept to have clear in your mind. Because actually a lot of the issues around mutations impacting on cancer derive from an understanding of that process. You can also have alterations in transcription or splicing, so a change in the DNA sequence changes how frequently the DNA is copied to RNA and then converted to protein. You may get more or you may get less of a particular protein produced. You can get amplifications or deletions of a chromosomal region. So you may get an extra copy of a particular gene. You may lose a copy of a gene, and this is passed on from cell division to cell division. You can also get much more serious or much larger alterations in the structure of your genome. For example, a chromosomal translocation, where one chromosome crosses over with another chromosome, say chromosome 2 to chromosome 5, and they swap part of the DNA sequence. And this can have a big impact in terms of the function of the genes on that chromosome. You can also get a gain of an entire chromosome or the loss of a whole chromosome. And if you look at the DNA in cancer cells within tumours, actually there's a huge array of changes in that DNA that are linked to a dysregulation of the control of DNA replication. There can also be changes in the modification of DNA. So your DNA isn't static, it's actually modified in a number of ways, for example by methylation. Methylation is one of the ways in which the body controls, or your cell controls, how active a gene is. If a gene is methylated, then the production of the RNA from that gene is suppressed a little bit. So that's another way in which gene expression is controlled. So there are multiple different ways in which your DNA can be mutated and changed, and many of these can impact on the development of tumours. What actually causes these mutations? So actually there are a wide range of potential causes. For example, exposure to UV and other types of radiation, for example, ionizing radiation from nuclear facilities. These can damage DNA, and actually it's a chemical process that occurs. The UV radiation hits your DNA and actually alters the chemistry of the DNA, changing the DNA sequence or resulting in a change in the DNA sequence. Other types of radiation, a good example of this would be radon gas. This is a gas which is naturally produced from certain types of rock. Radon gas is radioactive, and exposure to radon gas causes an increase in rates of cancer. If you live in Cornwall, where there's a lot of radon gas produced from the local stone, the rate of cancer is higher if you live there for a long time than in other parts, partly because of exposure to ionizing radiation from a radioactive source that just so happens because of an accident of geology that is more common in Cornwall than in other parts of the country. Other mutagens include free radicals produced during metabolic processes. So when you're burning sugars to produce energy, that actually releases a lot of energy, some of which can cause damage to your DNA. Viruses are a key cause of damage to DNA, and I'll talk a little bit about a couple of viruses that have been very closely linked to oncogenesis. And exposure to those viruses causes DNA damage, which dysregulates cellular replication, leading to cancer. And we can actually directly link those in a number of types of cancer and in a number of, with a number of specific viruses. Chemicals are a key source of mutagenesis. These are very well understood in some cases. For example, smoking. Exposure to carcinogens, which are found in cigarette smoke, increases the rate of mutagenesis in your lungs, in your throat, and in your mouth, which increases rates of certain types of cancer. And this was actually driven by epidemiology. People noticed many years ago that smokers have higher rates of certain types of cancer. And actually, it depends a little bit on how you smoke. So, for example, if you smoke cigarettes where the smoke is inhaled into the lungs, you're more likely to develop lung cancer. if you smoke a pipe, or if you smoke cigars where the smoke is concentrated in your mouth, you're much more likely to develop cancers of the mouth or of the windpipe. And so actually we can directly link exposure to these chemicals to the development of specific types of cancer. This is also true of asbestos. Some of you up by the library, if you walk past the library, you'll see there's a van there removing asbestos from the library building. Asbestos is a particular type, essentially a particular type of stone, which has very fine fibrous particles within it. If you inhale those particles, they go into your lungs, they increase the rate of mutation in your lungs. So exposure to asbestos, especially in its fiber form, increases rate of cancer. Individuals who were involved in mining asbestos during the 1950s and 1960s suffered a phenomenally high rate of cancer. Many of them died because of the work that they were involved in before we realized that exposure to asbestos could cause cancer. This is also true of certain types of food, although in many cases the direct link is much more difficult to ascertain because the effect is much smaller in size. Smoking and exposure to asbestos, and exposure to asbestos is a very good example of this. There's a very tight link between mutagenesis and the exposure to that mutagen. When we're talking about diet, actually it's an increase in risk rather than a definite progress to cancer. And this becomes much more difficult to understand in terms of the underlying mechanisms, but also in terms of the advice that comes through. And you'll have noticed this with newspaper reports recommending that you drink red wine or you don't drink red wine. You eat certain types of fatty foods or you don't eat certain types of fatty foods. Our understanding of that link is actually imperfect, and this is why nutritional advice can be so complicated. In addition to that, there are inherited issues in terms of copying and repair errors. And the BRCA1 gene that I mentioned as a risk factor for breast cancer actually is a very good example of that. BRCA1, the protein produced from the BRCA1 gene, is part of the DNA detection, damage detection mechanisms that allows cells to go, wait a minute, this DNA is damaged, we can't copy that, we can't continue with cell division. If you have a change in your DNA that you inherit from your parents that alters the function of BRCA1, you're more likely to develop breast cancer. And that can be predictive. So you can have your DNA analyzed and sequenced, and it can predict whether or not you're more likely to get cancer or not. And actually, you'll be learning a little bit about this in the Precision Medicine Practical later this week. The important thing to note is that all of these different mutagenesis events, all of the exposure to all of these mutagens, the accumulation of mutations over time can result in genome instability and increase the likelihood of further mutations. So having one mutation leading to two mutations and then leading to multiple other mutations increases the likelihood of developing other mutations which will lead to the development and spread of a cancer. So actually this is very well exemplified by a number of large-scale studies that have been carried out over the past decade or more, looking at the rates of mutation in specific types of cancer. This was a paper that was published in Nature actually a few years ago now. I think this was back in 2010. This was sequencing the DNA of small cell lung cancer and correlating the exposure of those cells to tobacco. So what scientists did is carried out a wide-scale sequencing of a number of small cell lung cancers from people who smoked and people who didn't smoke. And they were able to distinguish the genomes of individuals who had smoked from the tumor genomes of individuals who hadn't smoked. And so actually those mutagens and the different types of mutagens leave a lasting signature that can be detected decades after the initial mutagenesis event. So this study sequenced those genomes and it identified 23,000 mutations in a lung cancer genome. 23,000 mutations scattered around the human genome. And most of these were caused by exposure to chemicals in smoke. And it's estimated that one mutation is fixed, not just caused, but is passed on cell division after cell division after cell division for every 15 cigarette smokes. So that gives you an idea as to the damage that can be caused by exposure to tobacco smoke. I mentioned that there is actually quite a well understood link between certain types of viruses and risk of certain tumours. And so there are a number of different viruses where infection with that particular virus can be directly linked to the formation of oncogenesis and tumours. Examples of these include the papillomaviruses, which are involved in warts on the skin. These are predominantly benign and do not metastasize and spread to other tissues within the body, but also, more seriously, carcinoma of the cervix. And so infection with human papillomavirus, or HPV, especially in terms of the cervix in females, can cause the oncogenesis and can cause the initiation of carcinoma of the uterine cervix. And this link between infection with the virus, which infects those cells and causes DNA damage, which results in those cells dividing uncontrollably, has led to the development of vaccines which can prevent infection with HPV. And this actually is one of the ways in which we can proactively prevent incidences of certain types of cancer. And it's been very clearly demonstrated that vaccination against human papillomavirus prevents or reduces the incidence of carcinoma of the cervix. There are a number of other viruses that are linked to oncogenesis. Hepatitis viruses, which are linked to liver cancer. Epstein-Barr virus, which is linked to certain types of lymphoma. And these can be focused on certain populations in the world. So, for example, HPV is found worldwide. Certain types of Epstein-Barr virus infection are more focused on individual populations for a variety of different reasons. Now, these are all DNA viruses. So these are viruses that contain DNA within their genome, copy that DNA, and produce copies of the additional copies of the viral capsids. These do not integrate with the host genome. The damage that they cause is because of the disruption of cellular processes within individual cells. That leads to an increased rate of mutation, that leads to oncogenesis and the production of tumours. In addition to that, there are certain types of RNA virus. So these are viruses that don't have DNA in their capsids, they have RNA. This is then copied into DNA by reverse transcriptase and inserted into the host genome. And that insertion into the host genome can disrupt certain gene functions within the genome. This has been very well studied and in fact was initially identified as a class of virus through a study of HTLV, which is human T-cell leukemia virus, which is found predominantly in Japan and the West Indies, and of course human immunodeficiency virus, or HIV. And HIV partly came to the notice of clinicians because of an association with a very rare form of cancer, Kaposi's sarcoma. If you think back to what I was talking about in the very first part of the first lecture, sarcomas are cancers of connective tissue. And Kaposi's sarcoma, before the advent of HIV, was vanishingly rare. It was found in a very small number of very elderly individuals. In the 1970s and 1980s, young individuals in their 20s or 30s were being identified with Kaposi's sarcoma. And this was new. This was something that hadn't been seen in medicine before. What underlay that was that they had been infected with human immunodeficiency virus. What that virus was doing was it was resulting in the formation of a specific type of cancer. And this was partly driven by insertional mutagenesis. It was also partly due to alteration in immune system function, which normally suppresses the generation of Kaposi's sarcoma. And for these, actually, we have a very good idea as to the events deriving from infection with a virus and oncogenesis, and it's resulting in disruption of gene function and mutation. So I'm going to move on to talk a little bit now about the stages of cancer development. So as you can see from all that I've covered in the first part of this lecture this morning, there are a number of stages in the development of a cancer. You have the initiation event, where a mutation can cause a cell to undergo that initial disruption of cell replication, resulting in cell proliferation, promotional stage. This then can undergo further mutation or epigenetic changes, that's alterations in the regulation of your DNA, resulting in proliferation and selection, further mutations leading to further proliferation and selection, which eventually results in the progression of tumors. So this is the process that eventually leads to the growth and invasion of the tumor. And each of these stages links in to those hallmarks of cancer that I talked about in the first part of this lecture. So the initiation events, dysregulation of proto-oncogenes to form oncogenes, a mutation, for example, in RAS, where it goes from its normal function as a proto-oncogene to a mutated form or oncogene, which dysregulates cell division, followed by promotion and cell proliferation. That would be driven by alterations in suppression of growth inhibitors or resistance to growth inhibition and cell division and resistance to apoptosis, and eventually leading to tumor progression and metastasis and spread. In terms of the staging of cancers, there are a number of staging systems that are used in clinical practice to define what stage or how aggressive, how well advanced a particular tumor is. And actually identifying how far a tumor has progressed and what stage it is at is a key part of determining how to treat that cancer and what the prognosis is for the individual. In many cases, tumors are staged from 1 to 4, or using Latin numerals, 1 to IV, 1 to 4. And this is used for a number of cancers, for example, breast cancer, colorectal cancer. This is illustrated here. Stage 1, small size with no spread. Stage 2, moderate size with potential spread to lymph nodes. Stage 3 is large with a spread to lymph nodes. And stage 4 is a large tumours leading to widespread dispersal around the body. This is tightly linked to prognosis and 5-year survival. So the more advanced your cancer is, moving from that initial stage where the tumour might be quite small and hasn't spread throughout the body, through to stage 4 where the tumours are large and widespread, has a significant impact on survival rates over 5 years. Of course, the problem with this is that if a tumour is small and isn't impacting on normal organ function, you're much, much less likely to detect it. And so as you go through this staging, going from stage 1 to stage 2 to stage 3, the likelihood of detecting that you have cancer increases, but the prognosis decreases. And this is one of the key challenges faced in oncology today. There are other grading schemes as well. For example, the TNM scheme that stands for tumour, how far the tumour is growing locally, scored 1 to 4. Nodes, is there any invasion to lymph nodes, scored either from 0 to 2. And metastasis, has the tumour spread? And this is a binary count from 0 to 1. But you should be familiar with the fact, in particular, that the staging relates to the size and the spread of cancers, and that the larger and more widespread a tumour is, the tumours are, the less likely survival is. And this really is one of the other things that are highlighted in the first part of the lecture, was that there is a significant difference in prognosis and likelihood of survival depending upon the type of tumour you have, where it is originated, and the cell type that it is originated from. So for example, and of course this will be different between men and women depending upon the type of cancer. So for example, if you look at Hodgkin's lymphoma affecting the lymph nodes, 84% of individuals diagnosed with this survive over 5 years. If you compare that to lung cancer, the survival rate for lung cancer is much, much lower, down to 6%, with less than 10% survival. And actually, many of these cases are undiagnosed. So over 3/4 of these cases aren't diagnosed before they get to the point where there is a significant impact on life. Equally, this is true across women, but what you'll notice is that there are differences in some of the key, very common forms of cancer. And this derives from differences in organs which are present in males and females. For example, the uterus and cervix in females the testis and prostate in males. And actually, it's important to note exactly how common cancer is. These are statistics from the US. Every year, the Centers for Disease Control carry out a survey of the incidence of particular types of cancer and the mortality which is observed. Here we have estimated new cases for males and females, and again this highlights differences between males and females. The most common form of new case cancer in males is prostate cancer, with 174,000. This is for the US, out of a population of over 300 million. Whereas breast cancer is by far the most common form of cancer observed in females. In terms of estimated deaths, you can see that actually the most common cause of death linked to cancer is for lung and bronchus cancers. And these are the leading cause of death in terms of cancer. And this relates to the prognosis that I covered in the previous slide. We have got a lot better at treating cancer, and you'll be learning more over some of the following lectures, and you'll be learning a lot more next year about the ways in which we treat, diagnose, and manage cancer. But that has improved over time. If you go back to the 1970s, actually the median survival rate for cancer was 1 year. After diagnosis, your life expectancy on average was 1 year. And this has improved to, more recently, almost up to 6 years. These are figures for the UK. These do vary from cancer to cancer, so depending upon the type of cancer that you're diagnosed with, your prognosis and survival rate may be higher or lower. What is it that underlies this increase in survival? Part of it relates to earlier diagnosis. So if you go back to the staging of cancer, if you're able to identify cancer earlier, it increases the range of options that you can use. That may be surgery, that may be chemotherapy, that may be radiotherapy, that may be small molecule treatments. By screening, scanning, and looking for biomarkers, including genetic biomarkers, you can improve diagnosis and identification. Coupled to that, actually we have more and better treatments. Surgery, radiotherapy, and chemotherapy have been in use for many, many years now, but our use of those has improved and become refined. But we're increasingly seeing molecular targeting of individual cancers, often in combination with chemotherapy or surgery. And that improves prognosis. It improves the likelihood of getting rid of a cancer and it not coming back. It also reduces the likelihood that that cancer will spread. It's also important to note exactly how big an issue cancer is for health services. The example I'm providing here is for the UK, but this applies to health services across the world. And this is an estimate from a study carried out the year before last of the cost to the NHS of, actually this is NHS and private healthcare, of cancer. And what you're looking at is billions of pounds of expenditure. Treatment, diagnosis, and management of cancer is one of the major expenditures of the health service in the UK. And this is set to grow. This is a projection over the next few years, looking at where the costs of cancer are expected to grow. And at the moment, we're in 2019 now, it's just below 15 billion pounds per annum. And that's expected to grow and head towards almost 20 billion pounds. So improving diagnosis, improving care, and improving the treatments that we have is a major, major challenge for the NHS, and it's something that you will come across whether you're working in a community practice or whether you're working in a more specialised centre, because cancer affects all of the population. Everybody in the population has some kind of relationship to cancer. Either they themselves suffer from cancer or they know people who are affected by cancer. Many of the patients that will walk into your surgeries, if you're in a community practice, may have had cancer in the past or are likely to get cancer in the future. So understanding cancer and how it impacts on people's lives is a very important thing, not just in terms of the treatments that you may be handing over, but also in terms of how you deal with individual patients, understanding the challenges that they're going through. So as I say, one of the key areas in which we're improving is in cancer screening. This is both in terms of breast cancer with breast screening, smear tests, checking for cervical cancer, and looking for bowel cancer screening. And you'll see in the literature, both in GP surgeries, in hospitals, and also in pharmacies, that there's an increasing use of communication to try and raise people's awareness of the need to undergo cancer screening. And that screening can be critical. If a cancer is detected a few years early, it can result in much, much better prognosis. And the NHS has driven a lot of this, screening for bowel cancer from the age of 60, cervical cancer from the age of 25, and breast cancer in particular for women from the ages of 50 to 70, when there's the highest rates of incidence. As I said, there's also an increasing understanding of risk factors associated with cancer. These include age, genetics, and exposure to risk factors such as smoking, inactivity, obesity, alcohol, alterations in diet, and infections. And as I highlighted with the human papillomavirus, with hepatitis, and with HTLV and HIV, actually infections and viral infections in particular are linked to oncogenesis in a large number of cancers. So understanding that and intervening, and the HPV vaccine is a very good example of this, is a key way in which we can change the likelihood of people developing cancer, as well as dietary and lifestyle factors. And of course, it's also always important to note that this has changed as time has gone by. This is an advert from the 1950s for Camel cigarettes, where the Camel company made a point of noting that more doctors smoke Camels than any other cigarette. And this was at a time when really we didn't really understand the link between smoking and cancer. And it's instructive to see how much society has changed, to look at the way in which smoking is now handled as a society. So just in terms of summary, I'll leave you to read these at your leisure. We've covered what causes cancer, the ways in which cancer develops, and touched upon some of the ways in which one can intervene. You'll be learning more about this over the course of the next few days. There is a range of resources that you can access to learn more about this, and of course I'm more than happy to answer any questions that you may have. Thanks very much, Patrick.